And we're on. Good afternoon, everybody. Welcome to day 20. If you like your arbitrary numerical milestones in base 10, then off we go. So, yes, day 20. If you recall, yesterday we talked about the way that transcript level quantification works, at least roughly, kind of global top level view of how that kind of quantification works. And then we proceeded to install a piece of software that does that kind of quantification. So we installed Salmon using Conda. And then the last thing we did before we left for the end of the day was start running the command to build an index for Salmon from our downloaded transcriptome. So we downloaded the transcripts FastA file from GenCode, and then we used Salmon to build an index from that file. And that was where we left things yesterday. And with a promise that today what we would do was write a script to run Salmon over our 12 RNA-Seq samples, which are currently spread across 48 FastQ files, the FastQ files which we ran the QC on last week. So that's what we're going to try and do today. So I realised that a lot of the recent lessons have been fairly hands-off and have been quite a lot of listening to me prattle on a little bit about various concepts. Today is going to be a bit more hands-on, so today is much more about getting this script written and getting it running. So a bit more hands-on today, and then tomorrow we're going to turn our attention to R. So that's where we're going to be from tomorrow, is looking at getting R installed and running, and then trying to work with R to analyse this data that we've generated over the last that we've kind of downloaded and then generated over the last couple of weeks. So that's where we're at, and that's the plan for today. So the first thing I'm going to do is pull up my VM, which is running, and I'm sat in my home directory. I think my present working directory is my home directory, and all of my data that I've got is currently sat in my week 4 directory in my share. And if you remember, we started working yesterday in the week 5 directory because it's week 5. So that's where we started putting our work yesterday. So the first thing that I want to do is a little bit of housekeeping, a little bit of tidying, because the process of downloading the data in the more robust way that we did it, where we downloaded the SRA files and then converted those to FastQ files, has left us with quite a lot of data that we've downloaded but we don't necessarily need to keep, and that's the SRA files. So the first thing we're going to do is a little bit of housekeeping, and we're going to exploit the fact that we've got a week 4 directory and a week 5 directory, and we're going to move what we want to keep data-wise into the week 5 directory, which is where we're going to analyse it. And we're going to then discard what we don't want to keep from the week 4 directory. So the first place that I want to go is into my week 4 directory, which is in my share. So cd share week 4 to change my working directory, and check with pwd that we're in the right place, and then list this directory. And you'll see that I have got, for each of our 48 SRR sequence read archive accessions, we've got both a directory, and those directories contain the SRA files that we've downloaded, and we've got a FastQ file, which is a product of the FastQ dump conversion from those downloaded SRA files to FastQ. And that's the results of our get SRA robust script, which did both of those tasks. So used prefetch to download the SRA file, and then used FastQ dump to convert that SRA file into a FastQ file. So hopefully you've all got something fairly similar. If you haven't got something fairly similar, then the thing to do is to get this script, which is available on GitHub at the location that I showed you yesterday, and I will I'll pull that up for you in a second and just show you where that is. But if you get this script, and as long as you run this script in your share directory where you've got disk space, because if you remember, your virtual machine, which is your root partition, or the / in this df table, doesn't have very much disk space because we set it up with a very small disk. Now we can, in theory, we can resize that disk, and I've been looking into doing that. It's a slightly tricky procedure, and I don't really want to do it unless we absolutely have to. Whereas your share directory, so for me that's this line in the results of df, that can see whatever space you've got on the hard drive of your machine that your host machine. So on my host machine, I've got it's a roughly 500 gig hard SSD, which has got about 300 gig used. So I've got a couple of hundred gigs of spare space there. So that is a place where I can quite happily download this data, and it's fine. All of the FASTQ and the SRA files all together, you can see there, occupy about 34 gig of disk. So that's way more than we've got on our VM. Lots more than we've got on our VM. So that's our 48, because that's 48 SRA files and 48 FASTQ files. Even gzipped takes up quite a lot of space. So the chances are, if you run this script and it failed for whatever reason, it may well be that you're not working in your share directory. But for whatever reason, if at this point you want to catch up to make sure that you've got the same data that I've got, then you need to run this script, and you can get this script from GitHub. So let's just make sure we know where that is. So second, let me find the GitHub. There we go. Once you're at that repository, which should be publicly available, then you can navigate into the appropriate directory. So navigate into week 4, and then the script is get_SRA_robust, and you can get that script there and make sure that your script is the same as my script. You run that in your share directory, and you will get a copy of the data up to where we are now. Okay, so let's do our housekeeping. That was the first thing we wanted to do, which is we want to keep these FASTQ files, but we want to get rid of the SRA accession named directories which contain the SRA files. So what I'm going to do is I'm going to move the FASTQ files out of here and into week 5. So I'm going to move mv start.fastq.gzip. So that's all of the FASTQ files, anything that ends with .fastq.gz. So that's the first thing that we're going to do, and we're going to move those into /week 5. Okay, so we're going to go one directory up and then go one directory back down into week 5, and that's going to move all of our FASTQ files into the right place. So just check that that's worked. So get into the habit, make sure we're in the habit of checking when we've done a command that what has happened is what we expect to happen. So you can see my FASTQ files are all gone, and now what I've got left is the SRR run SRR accession named products of prefetch. So du my current directory now, I've got 15 gig, and du week 5, which is where my sorry, I should make that human readable, which is where the FASTQ has gone, and that's 20 gig. Okay, so that's the FASTQ has moved out of where we are now, and let's just check as well by listing week 5 to show you that the FASTQ files are all in there. So that's where our data is. So that's good. That's moved that data and made sure that when we run our rm command here, that we're not going to accidentally remove our data, which was the one thing that I was slightly worried about. So we want to remove all of these SRA files because we don't need them anymore. We can re-download them. We can rerun prefetch to download these at any point that we want. So it's not like we're deleting an irretrievable copy of this data. But for the foreseeable future, we're not going to need these files. They're not going to contribute to our analysis in any way. So let's get rid of them. So to get rid of them, we need to rm. We need to be very careful with this. So we're doing an R because we're going to recursively remove directories. Actually, let's do it slightly more safely. Let's do rm and then srr. So all of the directories start with srr, and nothing else in this directory starts with srr. So we can do srr and then star, and then if we just want to remove the SRA files, then we can do in each of those directories that match that first pattern, we can then delete any SRA files that we find by doing star.sra. And this will just delete the SRA files and avoids us having to do an rm minus r, a recursive rm, which if you type it wrong can be pretty destructive to your files. And it can be nice to try and avoid doing an rm-r if at all possible. So this will remove the SRA files and shouldn't refuse to do that. We can check. So if we just look in one of these directories, then we should be able to see, hopefully, that yes, it's empty. So that returned nothing. So the file that was in there has now gone, and that means that we're left with these 48 empty directories. And to remove an empty directory, we can do rmdir. And rmdir is cowardly and will refuse to remove directories with any contents, so we're protecting ourselves against sort of descending into directories and removing all of their contents. And again, we can use the wildcard, so srr and then a star with the wildcard system anymore. So those have now gone, and that's freed up 15 gig of disk space. It's always important to think about these things, and we didn't need those. We didn't need those files hanging around. So, and as I said, we can always re-download them. The other file that we've got in here that we will need for what we're doing today is the SRA run table. So that's the table that we got from the SRA run selector last week, which contains all of the metadata relating to our samples, relating to our SRA samples, our 48 SRA accessions. And we need that because we need to know which SRA accessions belong to which geo samples and so forth. So we need that information. So I'm going to move that SRA run table as well to week 5. So move SRA run table to .. so one up and then into week 5. To put that in the right place as well. So that's all of the tidying up that we need to do. So yeah, that's all of the tidying up that we need to do to be able to proceed with today's exercise of writing this script to run Salmon over these 48 files. Okay, so my current working directory now looks like this. Got a couple of scripts, and we've got the output of the QC that we ran last week. But we've got no FastQ files or no data in here anymore. So we're going to change our working directory to to week 5. Slightly more careful with my typing. There we go. So pwd. There we are. And if we list what we've got in here, so we've got our FastQ files. So list --1 star.fastq.gz pipe into WC-L to tell us how many lines there are, 48 FASTQ files, which is what we expect. Okay, so we've got our 48 FASTQ files. We've got our SRA run table, which is the metadata about our experiment. So that's got all of our SRA accessions in and a load of information about the experiment for those SRA accessions. And also importantly, for the purposes of what we're doing today, one of the entries is these GSM accessions. That's the geo sample accessions. So that tells us which of these SRA files, which of our 48 files belongs to which of our 12 samples. So if you remember, each of our 12 samples that we want to analyse is represented by 4 different sequencing files. And the reason for that is the construction of the NextSeq flow cell. So our sequencing experiment, and the SRA tells us this, the information that we've got from the SRA tells us that our sequencing experiment was done on a NextSeq 500. So that's an Illumina sequencer. It's been around for a number of years now. In fact, there's a new NextSeq platform that's been released, that was released in January this year. So the NextSeq 500 has now been superseded. But the NextSeq 500 is a really well-used sequencing platform. It sits between, in terms of output, it's when it was originally released, it sat between the MySeq, which is the sort of baby, or at the time was the baby Illumina sequencer, that's kind of benchtop sequencer, and the HiSeq, which is the kind of factory production line sequencer of Illumina, and it's a benchtop sequencer. It's capable of producing around about 400 million reads in a run. So it's a really good sort of mid-output sequencer, and it's very good for this kind of size of RNA-Seq experiment. One of the, it's not a limitation, but the thing about the layout of the NextSeq flow cell is it has 4 lanes. So it's run. The sequencing is run in these 4 lanes. So you get kind of four sets of files out at the end. So you get independent results from each of those 4 lanes. But the flow cell only has a single loading point. So unlike the HiSeq, which has 8 lanes, and each of those 8 lanes gets loaded independently, so you can put different libraries on each of your 8 lanes if you so desire. The NextSeq only has a single loading point, so any libraries that you put onto your NextSeq flow cell all end up on all 4 lanes. There's no alternative to it. And so that's why we've got 4 files for each of our samples, because we have 1 file per lane. So each of these samples has been sequenced on a NextSeq flow cell, and actually I would imagine that all 12 were sequenced on the same NextSeq flow cell at the same time. So you get 4 files for each. You get one file per lane for each sample, and that's why that's why we've got these 4 files per sample. Just to I don't know if this is working or not. I'm just drawing something on my whiteboard, and then we're going to look to see if I can spot it. Sorry, I've just taken the time to sync up my whiteboard, but it doesn't seem to be working properly. Because it just seems to have stopped working recently, which is a bit of a shame. It really doesn't seem to want to work. Anyway, I was just drawing an NextSeq flow cell. I do actually have one in my office, but my office is inaccessible to me at the moment, so there's not a lot I can do about that. But I just wanted to kind of outline the reasons for why we're trying to deal with this configuration of 4 files per sample. Right, so see a few people saying why have they got less data than me? If you don't have the same amount of data as I have, so if your directories are smaller than mine, there's a couple of things I would check. I would check byte for byte. So I mean, I can show you these. This is the size of these files byte for byte, and I can put a copy of this onto GitHub after the lesson if people want to check, because the summaries, this kind of human readable summaries, are not accurate enough. There's going to be rounding errors in there, although obviously a discrepancy between 22 gig and 30 gig is probably too big a discrepancy to ignore. So you can check byte for byte the sizes of the files. That's as close as we're going to get. I could also MD5 sum these, or SHA-256 sum them, and put those hashes up as well for people to check against, to make sure that you've got the same data. If you don't, if what you've got is not the same as mine, then if you rerun the prefetch script, if you rerun the get SRA robust script, it shouldn't do any unnecessary work. So if you run it in the same directory that you were in before you've done all this tidy up, and I'm sorry if you've done the tidy up and I'm telling you to do this subsequently, but if you run it in that same directory, it won't do any unnecessary work. So it won't re-download SRA files that are complete, for instance, but it will download them if they're incomplete. So it's worth running that again to check that it's got no work left to do. So I'll put after the lesson, I'll put the file sizes in bytes and the SHA-256 sums of the files up on GitHub along with today's script, and people can check that their data is the same as my data. But so what we want to do now is move on to today's script. So the first thing that I want is some input. So what we need to think about is we've got these 48 files, but they represent 12 samples. So when we run salmon, we want to run salmon 12 times. We don't want to run it 48 times. So not once per FASTQ file, but once per sample. And that's quite an important distinction. And it means that whenever we run salmon, we will be giving it 4 FASTQ files as input, the 4 files that correspond to the sample that we want to analyse. And so in order to do that, we need to think about how to get from our SRA run table, which has 48 entries, to a list of the geo accessions, which tells us what our 12 samples are, and then to get back from those 12 samples to which 4 files belong to each of the 12 samples, which is a little bit of, we've got to do a little bit of kind of gymnastics here, which will feel a little bit redundant, but I want to keep this, as much as possible, I want to keep this SRA run table as being our kind of reference point for this experiment. So what we're going to do is the first thing that we're going to do is we're going to cut the GSM accession numbers out of that out of our SRA run table. Okay, so that's the first job, and we're going to do this outside of the script. We're going to do this before we start writing the script. So this is just going to be a single command line. So the first thing that you want to do when you've got a table with a header line is remove the header, because we don't want to pull the header into our subsequent processes. So we're going to take off the header line by doing tail n+2, which prints us everything from the second line of the file to the end of the file. So that just removes the first line of the file. That's removing that header line. And then we want the column that has the GSM accessions in. So we want to cut. Our delimiter is a comma. And I will tell you from experience this afternoon, because I don't want to seek it out, that the field that contains the GSM accessions is the 19th field in our CSV file, in our comma separated values file. So cut -d, comma, -f19 will give us, for each row of our table, it will give us the GSM accession that's in that row. And hopefully what you can see is that each of these accessions is repeated 4 times. So you've got 378 4 times, 379 4 times, 380 4 times, and so on. So at the moment there's 48 rows here, 48 entries. Each one is so each of 12 things repeated 4 times over. We want to get rid of that repetition. So in order to get rid of that repetition, we want to find the unique entries in this list, which you can do with a Linux command which I don't think we've come across yet, which is unique. Okay, so unique will take a list, a list that you give it, and it will return you only the unique entries in that list. So only if anything occurs more than once in the list, it will just return you it once. So that will take this list of 48 things and give us back a list of 12 things. It will only do it if the list is sorted, because it will look for duplicated entries next to each other, basically. So it requires the list to be sorted. Now this list is sorted. If you weren't sure, then you could pipe the results of cut through sort, which would ensure that your list was sorted in some way and that the duplicates were pulled together, and then pipe the results of that sort into unique. So we end up with a command with 1, 2, 3 pipes in, so 4 different commands chained together. The sort here is probably almost certainly unnecessary, but this will give us a list of 12 unique GSM identifiers. Okay, we can just check that there's 12 by another pipe to WC-L to make sure that we're getting the right number of things out the other end. So we can use cat to read them in as input for our for loop in our script that we're going to write. So let's put these into a file. So let's put them into a file called geoaccessions.txt. Okay, so we're just redirecting the output of those 4 commands chained together by pipes. We're redirecting the output of that into a file, which we're calling geoaccessions.txt. Okay, so that has pulled those geoaccessions out of our table, and then if we can just check that that file contains what we expect it to contain, there we go. So GSM 44323 78 and then 79 all the way up to 89. So that is what I'd hoped to see. Okay, so that's what we're working with. That's the input for our script. So now we want to start to write the script, and as I've done previously, I'm going to borrow from earlier scripts to save me a certain amount of typing. So I'm going to copy a former script so that, in fact, I may not bother this time. Let me cat a script just so I can get the hash into my paste buffer. That's all I want to do. Week 4, or week 3 will do, doesn't matter. Get uniprot data. There we go. So I'm just going to borrow the hash from here. Okay, good. Right, so we're going to write our new script. So we're going to do nano, and we'll call it run salmon.sh, because that's the aim of the script. That's what we're trying to do, and we're going to build this script gradually as we go. So as with all scripts, we start with the shebang line, so hash !bin/bash to tell to tell the command line effectively the thing that executes this script, the executable on the system, which is going to run this script, is going to interpret this script. So this is a bash script. And then we want to get our input. So the geoaccessions file that we just created, we want to get that in as a variable into our script. Okay, so we want to read input into the script, and we're going to do it in the same way as we've done a few times now, using command substitution. So that's the dollar with the rounded bracket, that's the notation for command substitution, and then whatever command is inside the round brackets is run, and the output of that command is set as this variable geo. Okay, so we're going to cat our geo underscore accessions.txt file, and so that will give us those 12 geoaccessions that we just got into that file. It will give us those as a list of strings that we can then iterate over. So then we can write our for loop to go for I in $geo, and then it's do something done. Where the something here, to begin with, let's just echo $I. Okay, let's start by having it keeping it really simple and just echoing back to us our geoaccessions one at a time. Okay, so before we do anything else, let's check that this works. Okay, so Control O to save the file, and then Control X to exit the file, and then check that it's executable. So our file, oh god, yes, we've got loads of files in here. Our file is executable, and we know that because it's in our share directory. Okay, so rather than listing everything, let's just list the script. So there you go, there's the script. It is executable. It's executable by the owner and by the group. So we've got two X's in the first two positions where the X's go. We're a member of the group, the VBoxSF group, so we are able to execute this file. If you were doing this on a different system and you weren't running in a share directory where things would be automatically executable, you would make it executable by you the user, the owner of the file. So you would use chmod to change the mode of the file, to add execution permissions for yourself to that file. And then we execute it by telling bash where to find it. So it finds it in the current working directory, and then the name of the script is run_salmon.sh. And this will just echo back to us those 12 GSM accessions. Okay. So what we want to do for each of these accessions, we want to run salmon, and for salmon we need an input list of the 4 FASTQ files that belong to this sample. So how do we now go back to a list of, in this case, 4 things for each one of these 12 things? So how do we recreate those 48 rows in our file? And I thought about this today, and I've already written this today, so I did what I said I would do, and I took some time to think it through and to try and come up with a reasonable way of doing this. And we're going to build it step by step. So the first thing that we're going to do is we're going to use our old friend Grep, which might have been a while since we've written any Grep, but Grep you can use to find the matching lines for any given input pattern. So I'll just show you an example here. We'll pick this first one, okay, and we'll do we will do Grep, and then our pattern, which we'll put in single quotes just to be safe. Actually, you don't need to put it in single quotes. Let's live life dangerously. The pattern, and we're going to grep the pattern out of our SRA run table. And what this should do is it should return us the 4 lines in our SRA run table which contain that accession. And actually, it seems that they contain the accession twice. That's fine. That doesn't matter too much. It's still returning us those 4 lines. And the key thing is at the beginning of those 4 lines, there's 4 different SRA accessions, and these point to our SRA ID. These are our SRA IDs, and we have, for each of these IDs, we have a .fastq.gz file in our current working directory. So this will allow us to go from the GSM identifier back to the 4 equivalent SRA identifiers. And we can do it by, again, using cut the way that we have done in the past. Sorry, let me just kill that. So if we pipe this back into cut, again, we've got to tell it the delimiter, because the delimiter is the comma, and we want field 1, so the first field, that's the SRA identifier. That will give us our 4 SRA identifiers that correspond to this one geo sample ID, GSM accession. So that's our list of SRR IDs. And then what we need to do after, actually let's get these inside the script, and then we'll figure out what to do next. Okay, so let's go back to our script. So nano run salmon.sh, and we're going to run this. We're going to put this command that we've just written here, this grep command, into our script. So I'm just going to highlight it so I don't have to type it all out again. But instead of having a specified GSM sample identifier, we're instead going to use our loop variable to define the sample identifier. So let's go back into our script. So within this loop, each time the loop runs, $i takes the value of one of our GSM identifiers. So we want to run our grep here like that, but instead of having a specific GSM identifier, we instead want to use our loop variable, so that each time the loop goes round, each of the 12 times that the loop goes round, it uses a different GSM identifier, because that changes with every iteration of our loop. And we don't want to just run this command and have it print the results to the terminal, because that's not any use to us. We want to capture the results of this command, and to do that we're going to use command substitution again, like we did these 3 lines earlier, where we use command substitution to capture the results of cat as a variable. We want to do the same thing here. We want to capture the results of this grep piped through cut as a variable. So we're going to call this variable SRR, and we use the round bracket notation to do command substitution and to capture the results of that command as a variable. Okay, so that's in doing that as well, we lose the new lines, so we end up with a list of 4 things on a single line, which is what we want for the input for salmon. So that's convenient as well. So now if we, instead of echoing out just $i, if we now echo SRR, you'll see what I mean by that. Okay, so we're going to run this. This is the next kind of step of our script. So now we're using that GSM accession to get our SRR accessions back out of our file. So it's Control O to save, hit enter, and then Control X to exit, and then we rerun the script /run salmon. Okay, and hit enter, and you'll see we've still got 12 lines of output. But on each of those 12 lines, we have a list of 4 SRR accessions, and those 4 SRR accessions on each line correspond to the GSM accession that we're trying to process in this particular iteration of the loop. Okay, so that's one thing, but they're not those 4 things are not our FastQ files. So we need to introduce, for each one of these, we need to introduce a .fastq.gz after the SRR accession. Now this is where it gets a little bit trickier. We've seen a way of doing this before, of editing things like this before, using sed, the stream editor. And that's what we're going to use here. We're going to use sed. And the way that I'm going to do this Slightly convoluted, but it was the simplest way that I could come up with without doing a lot of awful kind of barbaric syntax. So what I'm going to do is I'm going to use sed to replace the spaces in here with .fastq.gz space. So that's what I'm going to do, and that will work for the first 3 of each of these accessions, of each of these lines, but it won't work for the last one. But the last one's easy compared to the others, and I'll show you what I mean by that in a minute. So let's go back into our script. Let's go back to our nano command and edit our script again. And so what we're going to do is we're going to use sed. So sed works on standard input, so we need to echo $srr and pipe it into sed, and then our sed syntax says, okay, we want to substitute, so that's an S at the beginning, and then the thing that we want to substitute comes after the first /. So we want to substitute the spaces in our SRR variable. Okay, then end that. So that's the thing that we want to substitute. Then after the second / comes the thing that you're going to substitute that with. So we're going to substitute the space with .fastq.gz space, because we still want the spaces between the different files. Then we want to end that. Now this would work for the first space on each line. But we want it to work for all of the spaces on the line. So to get it to work for all of the spaces on the line, we need to apply it globally, and for that we need to put a G at the end. Okay, just trust me, that's the way that it works. Okay, so that will apply that pattern globally. Okay, so that's our sed command, which is as straightforward as I could get it for the purposes of today. And again, we want to capture this output. We can reuse the variable name. There's no reason we can overwrite our existing variable with the results of this command. And again, we're going to do command substitution. So $ round brackets, then the command, and then close your round brackets, and that will capture the results of that sed command. And then after we've done that, we're going to check that the results are what we expect by again echoing out our variable. Okay. So there you go. That's our. That's what we're going to do next. So we're echoing the variable into sed, into the stream editor, changing those spaces and adding .fastq.gz in place of the space with its own space, keeping the spaces intact. And then we're going to echo that back out to check that what we've done is what we expect to do. So let's save our script one last time. No, not one last time. What are you talking about, Simon? We've still got things to do. And then let's exit. Okay, so let's check that this is doing what we think it's doing. Okay, so we're going to run the script, and now we've got, so because the other echo is still in place, so we echo the 4 things on their own, and then you see that the space is getting replaced as we expect with the .fastq.gz space, and that's working for the first 3 of our accessions, but the 4th one doesn't have the .fastq.gz on the end. So we need to add that to the end of the line. But that's relatively trivial to do. So in order to do that, back into our script, we just append it by effectively typing it onto the end. So you do srr equals srr dot fastq.gz, and that adds it on to the end of the whole. So you've got this string which at the moment ends at the end of the 4th SRR ID, but by doing this you just add that extra .fastq.gz onto the end of your string. Let's echo this one last time then. So echo $srr, save, exit, re-execute the script, and now we get a 3rd line of output for each one, and on the 3rd line we now have, this is what we need for Salmon. This is the list of our read files, which is what we need to pass to Salmon. So this is the input for our Salmon command. Okay, so that's what we were trying to construct, and I know that there's a lot of sort of messing about between various things in order to get there. And somebody said that this would be really easy to do in Perl, and I don't disagree. It would also be really easy to do in Python. That's what modern scripting languages kind of give you the ability to do this really easily. Bash is a bit more archaic and it may possibly in some ways a little bit more limited, but gives you access to these powerful command line tools. So I think we've done okay there. But this is just one piece of input for Salmon. So let's check what else we need for Salmon. So we installed Salmon yesterday. I'm in the right Conda environment. I'm in my RNA-Seq Conda environment. So Salmon quant is the command that we're going to run. So yesterday we ran salmon index. Today we're going to run salmon quant. Salmon quant actually has two modes of operation. So to get help for the mode that we want, we have to run --help-reads, which will give us the help file for the read-based quantification that we're going to run now. So this will give us help. The help file is very long. There's a lot of options, so I'm going to pipe it into less, just so we can see the top of the help file really easily. Okay. So here are the options. So what options do we need? And actually, they're all on the screen here. So we need to tell Salmon which library type we've got, and what we mean by this is what Salmon means by this is your library stranded or unstranded, and various other things. You can actually get Salmon to automatically detect what library type it is you're dealing with, and that's what I always ask for auto detection. So that's what we're going to do there. The library type for auto detection is a capital A, so that's what we're going to pass in there. We need to tell it where the index is. We created the index yesterday, so we've got that file, we've got that folder sitting on our in our current working directory, so we can point to that. And then it needs a list of, so we've got unmated reads, so we have single end reads, so it needs a list of files containing unmated reads, and that's the thing that we've just spent those lines of code producing, is that list of files containing the unmated reads. And then the final thing that we need is to tell it where to put the output. So where are we going to put the output? And we'll make the output the name of the geo sample, so the GSM sample accession. And then Salmon has lots and lots and lots of options, and we can scroll through pages and pages and pages of help here, because there's loads and loads of options to configure it for lots and lots of different ways of operating. Today we're not going to use any of these options. If you fancy trying to understand a bit more about how Salmon works and what it does, then read through these things because they're relatively informative. But for the purposes of today, we don't need to worry too much about the configurability of Salmon. We're just going to run it in the default mode of operation with our files. Okay, so I've got written down the things that I need. Let's type up our command. So we need to put this command inside our loop in our script. So let's go back to our script. There we go, nano run salmon, that's it. Right, so the command is salmon quant. Okay, so then we need to point to the index, and that is the index directory that Salmon created yesterday, which I have now forgotten the name of. So I'm just going to save and exit and list my current working directory. There you go, gencode _v33 _index. We haven't really looked at what's in there. It's essentially a lot of files produced by Salmon for the purposes of easily searching through that transcriptome for places where your present read matches that transcriptome. So you've got a bunch of files in there. Most of them are kind of binary format files, which Salmon uses to search. But the name of our index is the name of that directory. So gencode, in my case, gencode _v33 _index. Okay, so let's re-edit our script. Nano run salmon. and go to the end of the line and paste that in there. So that's the name of our index. We want the library type to be automatic, so -L, that's a lowercase l, not a 1, which is a thing that we run into a lot on the courses that we run, is you get a lot of errors when you write these scripts, and that's because people, it's in some fonts it's really difficult to tell the difference between L's and 1's. Actually in the default Ubuntu terminal font it's really not that bad. L and 1, I'll put the 1 next to it, look really quite different. But it can be tricky in some fonts to tell them apart. So it's -L, capital A. Then we want the output. So where are we going to put the output? And Salmon creates a directory for its output. So we're going to put it into a directory named after the GSM accession, which is available to us in our loop variable, $i. And then we need to give it our reads. So that's the final thing, is the reads that you want to process, which is -R. And we've constructed that in our SRR variable that we've created. So that, in theory, is the salmon quant command that we want to run. For each sample, it will be relatively quick, but it will probably take 10 minutes or so for each sample, because we've got probably between 10 and 20 million reads per sample to process, which is a relatively large amount. So it'll take a little while to run for each sample. So this is another thing where I will set this script running, and then hopefully our 12 samples will be quantified by the time we come back tomorrow. In fact, well, it definitely will be quantified by the time we come back tomorrow, but it'll probably take an hour or so to run this script. Okay, so that is our salmon command. And that will quantify each of our 12 samples. Even though we've got 4 files per sample, this will deal with those 4 files per sample elegantly, and will produce us 12 directories of output containing our salmon data. So I'm just going to get rid of these echoes because we don't need them anymore. So 3 lines to process from GSM accessions through to a list of FastQ files, and then a single line then to actually process those files using salmon. So that's the script, and as I said with the file sizes and the SHA-256 sums of the data, which I'll put on GitHub, I will also put this script on GitHub once the lesson is ended. So if you've missed anything I've said, if you've got slightly lost in the last 25 minutes, half an hour, then the script will be available for you to grab a copy of and run before we start to work with this data in R. Bear in mind that we're going to have a good couple of days, if not longer, of introduction to R before we get back to this data. So we've got, I'm taking the same assumption with R that I did with the command line, which is that nobody's used it before, and introducing it from scratch. So it's going to be a few days, maybe a week or so, before we get back to this data. So you've got plenty of time if you've fallen slightly behind today or in any of the recent lessons. You'll have plenty of time to make sure that this has worked before we start working with the data in R. And I can even probably make the data available if we need to do that. That will also be a possibility, depending on how people feel about that. Okay, so that's our script, and all that remains now today is to set this script running, assuming that this is going to work first time, which is a fairly big assumption. So let's see how this goes. So Control-O to save, Control-X to exit, and then execute the script. So run_salmon.sh to execute the script, and fingers crossed, here we go. Okay, so it hasn't immediately failed. That's the first thing, which is if it wasn't going to work, I would expect it to immediately not work, to do something that told me that it's not going to work. Since it's not done that, since it actually seems to be doing something, I think fingers crossed that this is actually working. So let's have a quick look at salmon is one of these commands that's fairly noisy. It tells you what it's doing as it's doing it. So there you go. There are the options that we passed. The command that we run, where our index is, what our library type is. Sorry, it's just started to produce output, so it's jumped me back down to the bottom. And then our list of read files. If it couldn't find any of those read files, it would have stopped at that point and complained. So it's obviously found those 4 files, which is good. And then it's loading in the index, and then it gets to the point where it's going to load in the reads somewhere. I think that's what it's currently doing. Actually, is it's running the reads. I don't know why this has suddenly started scrolling so slowly. There we go. So there you go, it's now processing the fragments. So it's processed 500,000 fragments, so for a million fragments now. Our index contained 227,063 targets, so that's the number of transcripts in the GenCode transcriptome. And this is now going to process our reads. So it's processed 1 million reads so far and getting on towards 1.5 million, I suspect, by now. So this will gradually chug through the reads in that first sample and produce us some output. If we look if we just open a second tab, we can see, so in my week 5 directory, we can see that we've got a directory now called GSM 443238. That's the first geo accession. We can have a look in that directory, and we can see that there's some subdirectories that have been created, including a log directory and some various other things. So Salmon is doing its work and is going to produce output. And at the end of Salmon running, in that directory we will have a file called quant.sf, and that file will contain our quantification estimates for this sample. And we can just check tomorrow that we've got all of those files before we start thinking too much about R. Okay, so we'll check that the output of this has been produced tomorrow. So there you go, we've got through 2 million fragments, that's 2 million reads in this library, and it's chugging away. So we will leave Salmon working, and we'll come back to it. We'll come back to the output tomorrow, and then after we've had a quick sanity check to make sure that our script has worked, I'll begin our introduction to R. Tomorrow we'll install R and a piece of software called RStudio, which is what lets us interact with R in a really meaningful way, and then we'll begin our introduction to R as a programming language. So that will be tomorrow. So tune in tomorrow for that, and I hope today has made sense. It's been a little bit more hands-on. As I said before, I'll make this script, the script that I've just written, I'll make available on GitHub. I'll do the work now to get that up on GitHub, and I'll also put a couple of files on there. One which will have the file sizes of my FastQ files in bytes, so you can check yours against mine. And I'll also have the SHA-256 hashes produced by SHA-256 sum of my 48 FastQ files as well, just so you can check that your data is the same as my data. And if for any reason your data isn't the same as my data, then you can rerun that get SRA robust script again. Bear in mind, as I said before, it's going to be a week or so, if not more, before we get back to this data. So you do have time to iron out any kinks, to let me know if you've really had any problems, and we can iron those out before we come back to work with this data again, once we've done our introduction to R. Okay, with that, I'll say goodbye, and I will see you all tomorrow, and stay safe, keep well, and see you online tomorrow. Goodbye, everybody.